Welcome to this lecture on biochemical engineering, the next lecture in the series. And what we're going to talk about today is receptor-mediated endocytosis, which we had been talking about in the previous lectures, and we'll continue that. Our specific interest or specific focus in today's lecture is going to be, as the title says, kinetics of receptor-mediated endocytosis. So what you see on the screen here is what we talked about in the previous lecture, which was the total process of receptor-mediated endocytosis. So just to recap, as I said, that it's very important for us to understand the process in order to be able to model the kinetics. So just to recap, as you can see over here, where the mouse is. So first, the first step, as is noted here by 1, that happens is that the receptor binds to the receptor is on the cell surface. Then next step is the receptor binds to the ligand to form this complex over here, which is denoted by C. Later on you will see. Then these complexes, this is for a certain type 1 receptor, these complexes diffuse laterally along the cell surface, so that the bottom part of it or the root part of it is coated by the coated pit. So the coated pit comes over here, and these coated pits are essentially a set of proteins, adaptor, clathrin and dynamin proteins, which gives mechanical stability to this receptor ligand binding. It allows, it's a function of this coated pit essentially to form coating when the invagination takes place, when the vesicle is formed. So this type 1 receptor, as I said, in which the receptor ligand binding first takes place, followed by the formation of the coated pit, as you can see over here. And then there is something called an invagination takes place. So part of the cell membranes, invagination essentially means, as you see where the mouse is, so the part of the cell membrane kind of invaginates, that is, goes in and traps the ligand. So the green particles that you see over here are the ligands, the black dots are the receptor, and these lines over here represent the coated pit. So they kind of invaginate and form a neck-like structure in which the whole thing comes in, and then this neck joins each other. Okay, the two neck join each other to form a vesicle. So what happens? What is the concept? What is the difference between invagination and a vesicle? Vesicle is something that is covered or sort of surrounded on all sides by the cell, whereas invagination is such that it is surrounded on most sides by the cell, but there is an entry or exit out to the outside atmosphere. Now what happens is that once this invagination process is over and the vesicle has been formed, then what happens is that the whole thing, the ligands are entrapped inside the vesicle. Okay, so then the coated pits get removed and they separate out into the different proteins and then go back into the to form coated pits again. So and then what you end up having is an uncoated vesicle, and then when the uncoated vesicle is formed, there is a pH change that occurs in the system. I have talked about this. In the pH change, what happens is the hydrogen ion pumps open up and they start to pump hydrogen ions inside the system. The reason this pH change occurs, or in other words the hydrogen acidification occurs, as a result of which the pH goes down, is so that when the receptor ligand binding is studied in the last chapter, there is a forward and a backward rate constant. So the forward rate is depressed or reduced and the backward rate constant is enhanced, as a result of which the receptor and the ligand dissociate. So whatever was in the associated or complex form, they dissociate into two and then separate out into two regions. The receptor go back, remain where they are on the membrane of the vesicle, whereas the ligands are swimming free. Then they separate out into this vesicle, breaks into two sort of vesicles. One contains the receptors on the cell surface and the other one contains the ligands. The ligands are either metabolized or thrown out of the body, and the one that contains the vesicle, they form these kind of channels and microtubules, and the microtubules leave the cell. So that is a process that happens, and the reason I am going back is because now our plan is to be able to model this entire process, and we looked at some of the things that we talked about, and we also talked about the disease familial hypercholesterolemia, which we said is very prevalent among South Asians, and the reason we said that we want to study this is because there are three different aspects that are possible. So we understand there is a receptor ligand binding. So from this picture, for example, we understand that if we are to do a kinetic study, of the system. So, this, as this picture shows, that so you have the receptor ligand binding, that is association, you have the dissociation of the receptor ligand, and there is internalization, and then there is vesicle formation. So, these processes are there. So, we have to study the kinetics of all of these processes. So, the point is that when we talk about disease such as familial hypercholesterolemia, as I said before, that these disease can happen due to several mutations, different kinds of mutations. So, essentially the disease is that you have this LDL, low density lipoprotein, in the blood, and this low density lipoprotein is supposed to go into the cells and supposed to be metabolized. Now, when you have the disease, this LDL remains in the blood, the ability of the cells to internalize it and metabolize it goes down. Now, the question that we have to probe is, why is it not being internalized, or why is it not being metabolized? Is it because the receptor ligand binding kinetics is there's something wrong with it? It's there's a retardation, or there's something wrong with the dissociation association kinetics, or is it the internalization kinetics, or is it the formation? What was the third point? Do you remember? The three mutations that we talked about. One was the association dissociation. One was the internalization, and what was the third point? The formation of the coated pits. We said, is there a problem with the formation of the coated pits or the lateral diffusion of the receptors to the coated pits? Now, in order to do that, in order to really quantify this process and understand that when a person has a disease, which is the one that is really the one that has to be taken care of, or the, something we can do. So, what we can do, the doctor might prescribe drugs which can alter, even so if it is a genetic disease, the doctor may be able to prescribe drugs which can alter some of these kinetics, okay, the reaction kinetics, some of these processes. But in order to be able to identify these, we need to quantify. The process of quantification that will follow in today's lecture is, first we need to come up with a model of our own, and which we think is a reasonably good model. Then we need to perform experiments, and then we need to fit our experimental data to our model to be able to evaluate the kinetic data. Now, what we do, we come up with our model, perform our experiments, fit our data, and then we perform our experiments for two different kinds of cells. We perform it for normal cells, we perform it for cells having familial hypercholesterolemia, and then we can compare the rate constants that we get for people normal cells and the rate constant that we get in cells having familial hypercholesterolemia, and maybe at the end of that we may be able to conclude some things that these are the constants that are defective, or this is a process, because it's important, you have a multiple three things we talked about, but even after three there are other processes. But at least for the ligand or the LDL to get into the cell, these are the three basic steps: the formation of the coated pits, if it's type 2 for example, formation of the coated pits followed by the receptor ligand binding followed by the internalization. And this is a sequence. These are not processes that happen in parallel, these are processes that happen in series. So, what is the disadvantage when a process happens in series? There is advantages also, but the disadvantage, advantage is that the driving force is, we can use the same whole driving force. The disadvantage is that when a process happens in series, if one of them, these processes go kaput, then the entire process go kaput. Okay. So, we need to figure out which is the one. So, that is our, to that end, let us work. So, these are the three mutations that we talked about. The coated pit and the receptor ligand binding and the internalization. So, this is the first thing. Okay. So, we are going to do a kinetic modeling on this. So, let me first describe the experiments to you, then I will come back and do the modeling, and then we will compare the experiments with the experimental results with the modeling data. So, this is the first experiment that we do is known as a pulse chase experiment. This experiment is called, as it is written in red, pulse chase experiment. So, this is a very common experiment that is done in case of LDL, and the fact that we exploit over here is that at 4 degrees centigrade, internalization of low density lipoprotein does not occur. Okay. The internalization part does not occur. So, what are we trying to do? We have a series of reactions that are happening. We are trying to freeze it down to a temperature where one of the processes is cut out, and you essentially have a couple of other processes staying there. Okay. Now, the coated pit, we will come to that a little later, maybe in the following lecture. How do you model the coated pit? Because when we talk about kinetics, let us understand this. So, we can talk very clearly about kinetics of the receptor ligand binding, because these are binding processes, right. So, you can talk binding and dissociation association process, you can talk about kinetics, you can talk about kinetics for internalization. But my question to you is, can you talk about kinetics for formation of the coated pits? So, it is a very complex process. Okay. First, there has to be the association of the three different kinds of proteins, they have to come together. So, definitely you can talk about some kind of kinetics there. But the thing that we are worried about is not the formation of the coated pits in themselves, but the localization of the receptors on those coated pits, okay, which has to happen through what? Through lateral diffusion along the cell membrane of the receptors or the bound receptor ligand bound complex. So, even if it is a type 1, type 2, whatever, either the receptor will, if it is a type 1, then the receptor ligand bound complex will localize to the coated pit. If it is a type 2, then the receptor alone. But it has to. So, it is not necessarily the formation of the coated pit from these three different kinds of proteins that we talked about, but the actual localization. So, how do we model that? That is the question I have. Do you think it could be modeled as a kinetic process, or is it a diffusional process? What is it? Strictly speaking, the answer is that strictly speaking it is a kinetic plus diffusional process, because the things are moving in the lateral direction, which is diffusion, but when the receptors reach the coated pit, it still has to bind to the coated pit, right. So, there is a binding process involved in that, or a reaction process involved in that. We will see later in the course how to do that, but right now we are not worried about. In the pulse chase experiment, we are trying to deal with the other two processes, one which is the association dissociation of the receptor ligand, and the third one, that is the internalization. So, in the pulse chase experiment, you have what is known as a pulse phase first. In the pulse phase, you reduce the temperature to 4 degrees centigrade, and as a result of which the internalization does not occur, and therefore, it is at 4 degrees centigrade it is possible to separate out the internalization process from the binding process. So, here the step by step mechanism of this experiment. So, cells are incubated with radio labeled LDL, basically you need some kind of label to be able to identify it, some fluorescence or something at 4 degrees centigrade. This period is known as the pulse period. Okay. Next, cells are removed, and solution is removed, and cells are rinsed at 4 degrees centigrade to remove LDL that is not bound to them. So, you basically allow the cells to bind as long as possible. So, incubate means for a very long period. You allow the cells to bind to them as long as possible and during the pulse phase, and when, so essentially what you presume that the binding will reach an equilibrium or a steady state. Okay. So, once this has been allowed, then you let the take the labeled LDL out and rinse it out. So, okay, so that, why do you rinse it? So that if there is any extra LDL which is not bound, but it is in the vicinity, swimming in the vicinity, could also be taken care of, removed, because the rinsing is not going to, remember, the rinsing is not going to remove the bound LDL. Okay, the LDL that is bound to the receptor, rinsing is not going to remove that. So, rinsing ensures that everything that is free, all kinds of free receptors, LDL receptors that are there labeled, are removed. Then we take the system, warm the system to 37 degrees centigrade. What happens when we warm the system to 37 degrees centigrade? You allow for the internalization process to start. Okay. So, the system is now replaced with unlabeled LDL. So, unlabeled LDL means you can still have the LDL, but it is not going to make any difference, okay, because our measurements are not going to change. Our measurements are going to be based on 4 degrees centigrade. Now, but still, why do you think we put add the unlabeled LDL? We are not interested in measuring those LDL, but we still want them to be there. Why? What happens when you have a system, and what happens if you raise the temperature of the system? Binding will? Why? Why should binding break down at just as soon as? Yeah, but you cannot say that binding will break down. Some changes will happen. It could dissociate more or associate more. You cannot say that it will necessarily dissociate. Yes, but to say it more simply, The answer is that as you raise temperature, the point of equilibrium itself will change. Okay. The equilibrium rate constants, the forward and the backward rate constants, both functions of temperature in different ways. As a result, the overall equilibrium rate constant is dependent on temperature, and as you change the temperature, it will change. I cannot say a priori whether it is going to change in the forward direction or the backward direction. Now, why do I add the unlabeled LDL? So that I make sure that the system is at equilibrium. So, first equilibrium is disturbed, I allow a next equilibrium to be established, but such that I do not count those cells, because my counting is based on what had happened at 4 degrees centigrade. Okay. So, then during this chase period, what we start to measure is, but remember, all the measurement is only for labeled LDL, because unlabeled LDL you cannot make any measurement. So, I just that's a peripheral thing, you know, that don't worry about that, what I just said, that I add in more unlabeled LDL so that equilibrium is measured, but in our calculations it can never come in, because we are only measuring labeled one that emit fluorescence. So, during the chase period, now what will happen during the chase period? So, the process of internalization kicks in again. Okay. So, now in the chase period you have binding as well as internalization, but what about the binding? Is the binding taking place? Binding is taking place, but we are not counting them. Yes. So, just as I said, you know, D, the point D over here, binding is taking place, but binding is taking place to unlabeled LDL, which is not being counted. Then internalization is the one that is now being counted. So, both binding, dissociation, that is association, dissociation and internalization, all these three processes are happening in the chase phase at 37 degrees centigrade, but because the labeled LDLs have been removed, the association is not being counted. What are being counted? What are being counted? Internalization and dissociation, that also is being counted. Okay. Because it's allowed to dissociate, but the association is being removed, that is the binding itself is being removed because there are no labels. Is that clear to all of you, the experiment step? Okay. So, next, that you know what I said just now, the appearance of labeled LDL in the medium is also measured to determine the LDL dissociation from its receptors. So, we are measuring both the dissociation and the internalization. Now, look at D and E, tell me what you can figure out from these two. So, disappearance of LDL from the cell surface is being measured, okay, and appearance of LDL in the medium is being measured. Yeah, exactly. So, D is the disappearance of LDL from the cell surface, E is the appearance of LDL in the medium. So, D minus E equals the amount that is bound to the that is being internalized. Okay. So, this is more or less the thing, and so you can, this is, you know, just a measurement. So, you can measure the amount of LDL that is being bound to the receptor and that is being internalized. Bound to the receptor you can measure because it is on the cell surface. So, D gives you not D, but D gives you the disappearance, but you can also measure the amount of LDL that is there on the cell surface. You can measure the internalization D minus E, and degraded also you can measure under the microscope, because if you have, if it emits fluorescence and if the LDL is degraded, then you know it will have a different texture below the microscope. You can measure that. Just once I talked about, you know, how to measure, remember how to measure live cells from dead cells under the microscope, and when you look at it, it's very clear when you color them, when you label these, and so the dead cells and the live cells, it's not that you lose them. So, what I'm trying to say is that when you label the LDL, so LDL that's active versus LDL that is degraded, these two have two different colors which are completely different from unlabeled LDL, because there are going to be some unlabeled LDL also in the system. The unlabeled LDL would not come at all under the microscope, but the labeled LDL, once it is it's active, that one versus when it's not active or degraded will have different colors. So, you can measure them also on the microscope. Okay. So, now let us look at the results of the experiment. So, kinetics of labeled LDL disappearance of first order. and rate constant that I get is k bar. So, what is this 0.257 or something I get, but let us look at this thing over here that I wrote, this arrows. So, this is K2 is a, so LDL is my ligand, R is a receptor, LDLR is a complex that is being formed, LDLI is a complex in internalized form, and LDLD is a complex in degraded form. Okay, that is the terminology, and that is what we will continue to use for the rest of this chapter. So, remember, LDL is for the ligand, LDR is a receptor, LDLR is a complex, LDLI is the internalized complex, and LDLD is a degraded. LDLI is the internalized complex or internalized receptor, both the same, and LDLD is a degraded ligand. So, if you look at it, so what we are measuring in at 37 degrees centigrade for that matter is the disappearance, right. So, the disappearance, as we said over here, D, okay, is essentially these two, because of the formation of the internalization and because of the going back or dissociation, right. So, that is what I can measure together. 0.257 is what I, then as we discussed, that we can measure the amount that is in the medium and subtract that out, right, and then we can isolate the internalization from the dissociation. Is it clear to everybody? Okay. So, once we do that, we figure out that this is what I get. So, 0.01 is the value that is of dissociation, and 0.247. So, out of 0.257, 0.247, most of it actually 95% or whatever that number is because of internalization, the rate constant, and the dissociation is very slow. So, which means that internalization is the one that is dominating as compared to dissociation. Okay. Is that clear? These steps are clear, right? That is the main step for kinetics here. I mean, I will go through that again. So, first here, binding of LDL receptor on cell surface, this is a second order reaction that happens, formation of the complex LDLR, and then it dissociates back. This is the first order reaction. Then this is again taken, LDLR is a complex, and the LDLI is the ligand in the internalized form. So, it could be either in bound, because within internalization there are two different parts. Remember, what is it called? Early endosome and late endosome, okay, or early vesicle, late vesicle. Okay. So, what is the difference between the early and the late? What is the difference between the, let us go back to the picture again. What is the difference between the early and the late vesicle, or the early and no, coated pit is not the point. Coated pit, it could be uncoated, but here, right here, I mean, why are you? Yeah, the pH. The pH of the system is the difference between the early vesicle and the late vesicle, okay, because early endosome and late endosome, so to say, because as you decrease the pH of the system, they dissociate, and I have said this many times already. So, whatever is the case, whether it is early or late, the total amount of ligand in the system is still the same, right. Between these two, say this one and this one, as you see on the picture, the ligand is still the same, because the ligands are being trapped in there. So, really it does not matter. So, when I say LDLI, what I mean is ligand in the internalized form here, which does not matter whether it is bound to the receptor or free from the receptor. So, and that forms a degraded LDL, or the endosome in the degraded in the lysosomes, which is LDLD, and this is the total process that we are interested in. Now, what are we missing over here? Is there something that we are missing over here? We are missing the localization to the coated pits, and that is a little more complex thing. So, what we will do is, first we will write the entire kinetic model, write our equations, write a solution and everything, and then add the coated pit part at the proper position. Okay. What do I mean by saying that a proper position? Yeah, because we have two types of class 1 and class 2 receptors. So, for class 1 kind of receptor, it will come at a certain point in the model, another kind of receptor, it will come before. Okay. So, that is why I said. So, basically what we are doing is writing the kinetic model as it is, and then we write the write this model for the localization in the coated pit added in. So, that we will do at the last thing. So, this is for normal cells. Now, what you have to tell me that into it, what do you think that you will, what kind of results will you get if you do it for the disease cells, the cells with familial hypercholesterolemia? So, that was the point, right? I said that we will do the experiment for both kinds of cells, normal and diseased, and then we will have a model and we compared that. So, we already have a model, as you see the picture over there. So, as soon as you write the equation, you essentially, not the equation, the reaction itself, you already thought of a model. So, these rate constants are based on that model. So, what do you think, what will happen if you take a disease cell? Okay, let us talk about first k bar that you have. Was that going to be higher or smaller? That is what you are going to measure. That should be an easy thing to answer. I think the other one that you are trying to answer is hard for you to answer. What you can easily answer is what will happen to k bar. Will it increase or decrease? Increase. Anybody else? Why do you think it will increase? What is the? That is very hard. I don't think you should try to answer whether k minus 1 will increase or k 2 will increase. That k minus 1 will increase, you are saying. That is hard to say. I mean, I would ask you not to venture that path. But what you can talk about is whether k bar will increase, overall k bar, because that is something you can only measure experimentally. Even I cannot tell, I mean, whether which one of these two is going to be higher. But overall k bar is going to, what will happen? Decrease or increase? How many? Decrease. So, k bar is essentially gives you the disappearance of the rate constant. Disappearance from where? From the cell surface, yeah, that in the bound form, that, yeah, here itself. So, this on the cell surface in the bound form. So, if this increases, then it is disappearing more. There are several possibilities. If it increases, then it is disappearing more, in which case, which out of k minus 1 and k 2, which has to be higher? Let us talk of the two possibilities. Yeah. So, let us talk about the two possibilities. One is, you talk in a disease cell, you perform the experiments and you find that k bar is higher, okay, say twice or something. So, in that case, you tell me that which one will be higher out of k minus 1 and k 2? K minus 1 has to be higher. Is it clear? That is an intuitive way of looking at it, but hopefully that is clear. If not, I will explain. K minus 1 has to be higher, because if it is increasing, which means that this whole thing is not being internalized, it just leaves the cell. And if it is decreasing, then k 2 is smaller, yeah. So, that is, so let us see now what really happens. So, this is what really happens. So, for a disease cell, it turns out that everything is similar, and the second option. So, k 2 for the disease cell is smaller, okay, suggesting that the mutation of the LDL is not for this mutation LDL receptor is not internalized. So, what is the reason? So, all these possibilities were there. Remember, we talked about the three different possibilities. So, forget about C, because we do not have C in our model yet. So, there are two possibilities, right. So, that is why I said there are two ways of looking. There are two things that can happen. One is that Kd increases, or in other words, the k minus 1 increases. Okay, that is a possibility, that though it is binding, but the dissociation rate constant in the disease cell suddenly goes up. As a result, though it is binding, it is leaving, it is dissociating again. Another possibility is that yes, it is binding, the dissociation rate constant is not going up, but what is happening is the internalization rate constant is going down, and this is something that you and I cannot predict without doing the experiment, that which of it is actually happening. So, but my job was, what I was trying to tell you is that, okay, let us try and convert those possibilities in terms of mutations to kinetics, okay, because that is the way, when you have to look at physiological systems, that is what you have to do. What is first, those three things that we said over here, that is called, this is a slightly different way when you model a physiological system. This is the approach that you need to take. First is, you make a hypothesis. This is called a hypothesis, Okay. So, that is any physiological system modeling, you have to start with a hypothesis, unlike in a engineering modeling. Okay. Why? Because the systems are very complex. So, you have to come up with some hypothesis, simplifying hypothesis of your own. So, just like we came up with these 3 hypotheses here, and then you perform. So, this is the one and only way of doing it, there is no other way. Then you write a model or create a model, then you perform experiments, and then you check with your experimental data to your fit your experimental data to your modeling or whatever, and then check through finding out the rate constants whether your hypothesis was correct or not. Then if it is not correct, then what do? You get rid of those hypotheses, come out with a fresh set of hypotheses. That is the way people do when we do physiological modeling. You have to go in circles, there is a do loop out there. So, you make a set of hypotheses, write your model, perform your experiments, get your kinetic data out of it, and then check if your hypothesis is correct or not. If it is not, then you start with a fresh set of hypotheses and go in this circle till your hypothesis and your solution matches. Okay. So, this is the very the best way of doing physiological modeling, unlike in chemical engineering modeling, where you can just write an equation and then solve it. In physiological systems, you cannot do that. So, that is what we did. We had this couple of hypotheses. Now, we had these 2 possibilities based on this hypothesis. So, possibility 1 confirms hypothesis 1 and possibility 2 confirms hypothesis 2. When we actually got the data, we found that it is not hypothesis 1, but it is hypothesis 2 that is correct, which means that in this process of let me go back, in this process of this entire process. So, the part that is not working, okay, the part that is not working when a person has familial hypercholesterolemia is the internalization process. Okay. It is not the binding or the dissociation, they are fine. When I say that South Asians are genetically predisposed to familial hypercholesterolemia, what is meant is that it is not that their binding or dissociation of the receptor ligand that is a problem, but it is an internalization process of the receptor ligand complex that is not happening. Okay. So, now I will show you the experimental results here. Here this is the experimental result. So, as you can see in this tiny picture out here on the top, this one I will come back later, but let us look at this picture out here on the top. So, what is seen is that normal cells have fractional amount bound is much less with time. So, at t equals 0, at t equals 0, you have, say, this is fractional amount bound, the total amount bound is the same for normal and FH, familial hypercholesterolemia cells. But as time increases, the normal cell starts to internalize. So, which means what? That if these two are same at t equals 0, t equals 0 is the end of the pulse phase and the beginning of the chase phase. That is my time, that is when the clock starts ticking. So, excuse me. So, if these two are same at t equals 0, what does this mean? It means that there is absolutely no problem with the binding. Okay. Same amount has bound both to the normal cell and the disease cell. Okay. Now, when you look at this thing on the top, the familial hypercholesterolemia thing, it sees that the same point it starts, but the slopes are completely different. As a result, the slopes have to do with the kinetics, the rate constant of internalization, which means that slopes are different. So which means that the internalization rate constant for familial hypercholesterolemia is much lesser than the internalization rate constant for normal cells. Okay. As a result, what it means is whatever is being taken in is not being internalized. Fine. Now, this is the overall picture with time. So, the amount that formed is general, this is both for normal and for FH, this holds this picture that you see over here. So, what happens is a bound form, and I will show you, we will do the modeling in a minute. Maybe I will do that modeling first and then come back and talk about this. Yeah. So, just a sec. Yeah, this is the full model. Yeah, let us do the modeling for the. So, I will come back to this picture after we do the modeling. You will be able to appreciate this a little better, because these three. So, when we do the modeling, so this is our kinetic model, as you can see on the screen. LDL plus R giving LDL R, LDL I, and LDL D. So, that's the thing. So, what kind of simplifications can you make for the? So, right now we are doing the, as you can see, it's written there, kinetic model for the experiment. This is not the full model. The full model I will come to in a few minutes. So, this is the model for the experiment. So what kind of simplifications can you make to this model? What kind of simplifications can you make to this model for the experiment? You should be able to tell this. I mean, this is very easy. We already went through the steps of the experiment. What should be the steps? What should be the simplification? Last one. Last one I can leave out, but that's not what I'm hinting at. What is this pulse chase phase? There is no association because we have done it that way, right? We removed all the LDL that was nearby, all the labeled LDL that was nearby through rinsing. As a result, thing is not there. Okay, so in the chase phase, the K1 is 0. The pulse phase, the K1 was the only thing that was there, and a certain amount of LDL was formed. So essentially, I have to real R being the initial condition is that LDL-R is not 0 at t equals 0, LDL-R has a certain amount, okay, just as you saw over here. So LDL-R is not 0, so this is the beginning of the chase phase. The LDL-R is not 0 at t equals 0, it has a certain value. If you did a full model, then what will happen? LDL-R would be 0 at t equals 0, okay, but because we are separating out in the pulse and chase phase, and the value of LDL-R is whatever has bound, fine, whatever is bound. So there is no association. We are also aware, because we already performed the experiment, that K minus 1 is much less than K2, okay. So that the backward rate constant is much smaller than K2. So essentially what we can do, we can chop off this part itself, because first is K1 is 0 in the chase phase, and K minus 1% 3%, I don't know, 4% maybe, 4% of K2. So you can neglect that, and essentially you just have this part, okay. So this is my model now. Let's go through it one by one. So and DDT, so essentially I am writing because there is no, these are, why is there no diffusion? Because these are fixed to this localization to the lateral diffusion will come in is during localization to the coated pits. So DDT of LDL-R equals minus K2 N LDL-R, okay. DDT of LDL-I equals K2 here formation K2 N LDL-R minus K3 N LDL-I, and DDT of K3 LDL-I equals DDT of N LDL-D, this one, the formation equals K3 N LDL-I, and at t equals 0, as I said, LDL-R is not 0, it is given by some initial value, which is the value that is obtained at the end of the pulse phase, and excuse me, the internalization also is given some value, but it could be 0. You can assume it to be 0 because you have taken the cells to be at 4 degree, but the reason that is put over here is in case there is some residual internalization, that is before you froze it to 4 degree, if there is something that had already internalized and it is there, okay, so that you can measure if that is possible. Otherwise this could be 0, and of course an LDL-D equals 0. Is the initial conditions clear? So this one is very important, and these two could be 0, both of them could be 0, fine. So how will you solve this now? Set of equations. Yeah, that's one possibility. The other one is you can solve all three together also using the matricial method, yeah, whichever. But what you are saying is a lot easier. So this is, this is a very classical, this set of equations that we have is a very classical equation in physiological systems and also in some chemical engineering systems. Can you predict what kind of, just look at the equation there. Once you solve equation 1, put it into equation 2, what kind of solution you would get, especially let us say for equation 2? What kind of response you would get for NLDLI? What is that called? What is that kind of solution called? Biphasic solution. Okay, it's called biphasic solution. It's a very classical thing in most physiological systems you'll find, and even in many chemical engineering systems also, when you have these series kind of, you have essentially series kind of reactions. that is what you find, and A going to B going to C, it's a kind of thing. So I think, yeah, this is the solution that you get. So the first one is exponential. Now once you put this exponential into the second one, you get this exponential minus k2 minus k3. So this is the one, these two terms, this negative term over. So n plus n LDLI, not exponential minus k3. Now this could be 0, n LDLI could be 0, okay. And despite that, you will get this peak, because there are two terms which are opposing each other, and that happens because you plug in the first one into the second one. So these two exponential oppose each other. So when this one leads, then it increases. The first term k minus k2 leads, it increases. It reaches 0 when these two are equal, that is, and then it goes down again. So that is how you get the biphasic response. And then LDLD, you integrate this one more time, okay, then you will get this. Now what kind of response do you expect for this one, for an LDLD? Let us see one by one. So this one, t going to 0, n LDLR, t going to 0, it is n LDLR naught, okay, and t going to infinity, it is 0, okay. So it decreases from a certain value down to 0. This one, t going to 0, it is 0, okay, leave this term out, n LDLI naught is assume it to be 0. So this is 0 at t going to 0, t going to infinity is what? 0 again. So essentially it starts at 0, goes through a peak and goes down to 0. So here again this term is 0, okay, do not worry about this term. Let us talk about the rest of the terms here. So at t going to 0, this is 0, right, and t going to infinity, this is minus 1 over k2 minus 1 over k3. So essentially it flattens out, it saturates, it starts at 0, it saturates. Now let us go back to the plots, and this will make more sense, okay. So as you can see, this is the bound one, okay, as we said that it starts at a certain value, and these are values from the experiment. So this corresponds, so the model we did, and then we measured the values for the experiment, and then you can fit this. So the thing that is important over here is to have the model which have a sort of a model which fits the experiment, because what I am trying to say over here, these are not model results, by the way. What you see on the screen here, these are not model solutions, these are obtained from the experiment directly, okay. And now the good part is, that the lucky part is rather, is that we have a model which fits this qualitative nature. So whenever whenever you are doing this kind of physiological modeling, the first thing that you have to look at is that, see, why, how is physiological modeling different from normal chemical engineering modeling? The reason it is different is because the levels of complexities are very, very high in physiological modeling. So in a chemical engineering modeling, you can nail it down to the last diffusion term or the convection term or the reaction term, okay. So you can write an equation, a diffusion convection reaction equation, say, or some kind of reaction equation, and be happy about it, because you can write an equation and say, oh, okay, I think I put in all the phenomena in there, and if I simulate this equation, I can get the real thing, okay. Whereas in a physiological system, it is so complex, and it is so complex that we do not even understand these systems completely physiologically, okay. So the problem that we have over here is that it is so complex that we can forget about modeling this from the first principles, okay. You can put in, you can say, okay, I think there is some diffusion in there, you can say that I think there is some reaction in there, and put this in, but a host of things are going on, even these systems that I talked about, a huge set of things are going on in these systems that we are not even accounting. What about, for example, the reactions between the different proteins that form the coated pits? What about the reaction of the ligand to the coated pit as it is binding? Okay. What about the reaction that happens when the coated pit shreds its, the vesicle sheds the coated pit? What about what kind of reactions that happen, or dissociation that happens when the pH is decreases? So if you want to do full modeling, then what you have to do is you have to model for the ion pumps that are there, the hydrogen ion pumps that are there, and model that process itself, the process of pumping in the hydrogen. And so what I am trying to say is that the process is very, very complex, and you cannot even dream of actually doing it completely right. So what you have to do, you have to be able to pick out the basic things correctly, A, and B, more importantly, what you have to do, you have to look at the experiment and see that what kind of qualitative model will fit the experiment, that is another thing, that is something that you have to do. Excuse me. So here, as you see, that in the bound system it starts at a certain value and goes to 0, as we said. This one starts at 0, and so it starts around 12 nanogram per of LDL protein per dish, per petri dish. It starts at 12, goes to 0 in about an hour, and this one starts at 0 and reduces to a very small value in couple of hours, but it will still take probably another couple of hours to actually go to 0. And the degraded one, as I said, it starts at 0 and it should saturate out, and in 2 hours it still hasn't saturated out, as you see that it will still continue. The reason being that what is the reason? The process of all this means that the rate constants of degradation is very small, or in other words, the process of degradation is small, is slow as compared to the process of internalization or the process of binding. So as you can see here, the process of binding is the fastest actually. The process of internalization follows that. It is not as fast as the process of binding, but one of the limb is almost parallel to this, so it is reasonably fast, and then the process of degradation is the slowest. Okay. So this is the model we have for the experiment, and so next thing that we are going to do now is do the full kinetic model. So the full kinetic model is going to be a slightly more complicated, because we have checked out, here we had completely removed this section. So we need to add that section in, and let us see what happens. So this is what you get, and what comes in here is the NR, okay, which is the amount of free receptors out there. Okay. Here, as you see, NR being here, and this is the amount of free receptor that is there. So and C LDL is also there. Now what, as we made an assumption, remember, in the LDL receptor ligand group binding chapter, that you can assume that ligands are in plenty, which is something you can more or less assume, because ligand is in your hand, you put the system even in the body. So if you take a lot of protein fats, the ligand is obviously is excess. So if you want to do this experiment on a live patient, it's not possible, but if you still want to do, you can still pump in a lot of ligands, so the ligand is in excess. So you make that assumption here down there. So CLDL equals CLDL naught. That is the initial concentration, remains as the initial concentration because it's in excess, and you have NR over here. Now this equation as it is not solvable, but what we do is we use the constraint nRT equals NR plus N LDLR. So if you use the constraint, then you can replace NR over here, nRT minus N LDLR. So essentially this first equation is a second order equation with 3 unknowns. So what we have done, we have converted the second order to a first order by assuming that the ligand is in excess, A, and B, we have got rid of so we got rid of one unknown and we got rid of the other variable by using the constraint equation. But remember, these two, both the constraint equation and I have not said this in other cases before, but it is a lot more important to note that here both the constraint equation and this assumption are essentially assumptions. Okay, even the constraint equation is an assumption, and we will do that in the next lecture, why that is an assumption and what things we can do. So essentially it is an assumption. Why is it an assumption? Because, as I said, I think I discussed this a little bit in the previous class, that the number of receptors are being regulated, and it is not possible to assume that the number of receptors is a constant on a cell at all points of time. Okay. So once we do this, then again you get a similar sort of thing, similar sort of equation. So 1 minus exponential kt, and this should have been k is k minus 1 plus k2 over k1. So basically you substitute that over here and fiddle around with this a little bit, and so then you will get this k2 plus k minus 1 over k1. That would be your rate constant, and this is what you get for LDLI, and this is what you would get for the degradation. Okay, because see slightly this is different from, let's go back to what we got over here. look at the LDLI thing that you have over here, and here it's slightly different because this expression is, that the expression that you get over here is different, and when you plug that back over here, I mean, you can go through the calculations at home. So this is what you had before, is the same expression out here, but because this expression itself is different, so then you get a slightly different expression. And I think the limits are still the same, because as, let's see, as t goes to 0, this is no, the limits also have changed, I think. This goes to 0, and this goes to 1 over k3, right? And as t, and this one, because there is a 1 out here, when you integrate that, you get the t minus 1 minus exponential part. So let's look at the limit. So limits of this is, as t goes to 0, this is a certain value, and t goes to infinity, this goes to 0. That's fine. This one is, as t goes to 0, these two balance out, and you get a 0 out here, and then you get 1 over k3 out here at the initial point, and then you get, see, one of the differences that you had, and it's not mentioned out here, is that there we had taken an initial value, okay, for all of these, but what would be my initial value over here? So here, for example, an LDLR is 0 at t equals 0, and there we had an initial value for LDLR, the reason being that that value was what we got at the end of the pulse phase, right. So, but here that's not the case, we start with a clean slate almost, so you have the 0 value and because you have that change, all the rest of the thing kind of keeps changing, and this one is still the same, and then this one will change a little bit. So t going to 0, let's see, this goes to 0, this term goes to 1 over k, this term goes to 1 over k3, this term goes to 1 over k3. So definitely it's not 0 at t going to 0. At t going to infinity, it's 0 t equals 0. This is 0. This 0 at t equals 0. But at t going to infinity, it saturates out like last time to a certain value. Okay. So now let's look at the plot. Okay. So what is, the difference between this and this is that, as I said, that it doesn't go to this one, for example, at t going to infinity, this is not going to 0. Okay. Earlier, if you compare here, at t going to infinity, this went to the LDLI I am talking about. LDLI went to 0, but this is not going to 0. And so one of the things that happens is, even though this continues to be a biphasic response, but the biphasic thing is not similar to the biphasic that we had before, or in other words, it increases and then it doesn't go to 0, the internalization doesn't go to 0. The binding saturates out, okay, because why does the binding saturate out? Because the system, the equation, reaction attains equilibrium. Yeah. So the binding saturates out. The degradation keeps increasing, but can you comment on why? So these are, for example, experimental values, as you see over here. K2 is 0.2 minute inverse, K3 0.02, and so on. Can you comment on why the internalization does not decrease?
Because we have plenty of ligands, we have assumed, so bound is always there. Because the bound is always there, so internalized will always
Yeah. Whether we have plenty of ligands or not, that's not the question, but the point is that the binding, unlike in the last case where we stopped the pulse phase and prevented all kinds of binding to happen beyond the pulse phase, here the binding is a continuous process, right. So the binding goes on, and because the binding goes on, the internalization goes on. So we have run out of time today. Next class, maybe this is the first slide I'll show, and then we'll continue to do more complicated modeling about the coated plates and the general modeling, and we'll finish this chapter in the next lecture. So thank you.